We will here have a look at UMAP, which is a similar method to the t-SNE method that I covered in the previous video. UMAP stands for Uniform, Manifold, Approximation and Projection, which is a dimension reduction method that captures both local and global structures of high-dimensional data. The UMAP method was published in 2018. I will first show some applications where UMAP can be used and compare it with principal component analysis. Then we will have a look at a simple example that explains the basic math behind UMAP. We'll first see how UMAP and PCA perform on a classical MNIST dataset, which contains thousands of images of handwritten digits. Every row in this dataset contains pixel values between 0 and 255 for each image. The first column shows which digit the image contains. For example, the first row represents an image of a handwritten 5, whereas the second row represents an image of a handwritten 0, and so forth. Each image is a square that contains 784 pixels, which means that each image or row has 784 columns, in addition to the first column. Suppose that we like to generate an image of the data that is located on row number 6. We would then put the elements in column numbers 2 to 29 here, then the next 28 elements here, and so forth, until we have a square matrix with 28 rows and columns. This image is supposed to represent a handwritten 2. We'll now use UMAP and PCA to compress these 784 columns into just 2 columns and plot these columns in a 2-dimensional plot. We will then label the points in the plot so that we can see which point that corresponds to a certain digit. Similar to PCA, UMAP is an unsupervised method because it does not use these labels in its computation to separate groups or clusters. This is an example of a UMAP plot of 10,000 images in the MNIST dataset. Each point in this image represents one image. For example, this point represents the following handwritten 8, whereas this point represents a 4. We can see that the UMAP separates the images of the 10 digits quite well. In comparison, this is the corresponding PCA plot of the MNIST dataset, which fails to separate the digits. I will explain why in a minute. UMAP plots are commonly used in single-cell RNA sequencing to visualize high-dimensional gene expression data in 2 dimensions to identify clusters of cells with similar expressed genes. Each point in this UMAP plot represents a single cell. Points in the same cluster can be seen as cells that have a similar gene expression profile. Such a cluster of cells therefore usually represents cells of the same cell type. For example, the clusters may represent different cells in our immune system. If we were to use PCA on the same dataset, the clusters would not separate well. So why is this the case? Well, one reason is that a PCA plot usually only shows the first 2 principal components, because these explain most of the variance. By illustrating only the first 2 components, which in this case only explain about 5% of the total variance, we lose a lot of information that is stored in the other components. For example, we can see that cluster 2 and 6 are not well separated based on the first 2 principal components. If we instead plot component 1 versus component 5, clusters 2 and 6 are now well separated. The UMAP method does not hold important information in several components like this. Instead, it includes all information in just 2 dimensions, which explains why the UMAP plot better identifies clusters that exist in the high-dimensional data. However, the UMAP method is computationally expensive, which is why it is sometimes combined with PCA. One can therefore compress the variables by first using PCA and selecting, for example, the first 30 to 50 components that store most information, and then computing the UMAP based on these components instead of all variables. Another reason why PCA might fail to separate existing clusters is that it is a linear method. For example, if we were to project this non-linear two-dimensional dataset into one dimension, the clusters will not separate well with PCA, whereas the UMAP would do a good job in separating the existing clusters because it preserves local non-linear relationships. Although these points are fairly close in the global geometry, UMAP tries to preserve also the local structure. Close points are assigned a high connectivity weight, indicating a strong likelihood of being neighbors, which means that also these points have a high connectivity weight and so forth. A UMAP is controlled by these two hyperparameters that are set by the user. The parameter nNeighbors can be seen as the number of neighbors each data point selects for its local neighborhood in the high-dimensional space. The effect of this parameter is shown on the MNIST dataset. A small value focuses more on local structures, which might fragment some clusters. A larger value focuses the plot more on the global structure, where clusters may overlap a lot. The default value of 15 is usually a good value to use that captures both local and global structures. The parameter mean distance controls the minimum distance between points in the low-dimensional space that I will explain later on. A low value results in tight clusters, whereas a too high value might cause overlapping clusters. Setting these two parameters to 15 and 0.2 gives a nice UMAP plot based on the MNIST dataset. The parameter spread was not described in the original UMAP paper, but has been included in several implementations to control the distance between the clusters. Increasing the value of this parameter increases the distance between the clusters in the MNIST dataset. We will now discuss the basic math behind UMAP. The math that I will show comes mainly from Appendix C in the paper, where they explain UMAP in a similar way to the t-SNE method, which is a lot simpler to understand. We will use a similar three-dimensional dataset as in the video about t-SNE, and use the same random initial coordinates in 2 dimensions. UMAP may either use initial random data points like this, or points initialized based on the method of spectral embedding. We will here see how UMAP reduces the dimensions of this high-dimensional data into low-dimensional data. In other words, how it reduces 3-dimensional data into 2-dimensional data. In comparison to t-SNE, which uses a Gaussian-like distribution that I explained in the t-SNE video, UMAP uses an exponential kernel based on some distance metric. I will here use the Euclidean distance. UMAP uses this equation to compute a symmetric matrix, whereas t-SNE uses this equation. UMAP starts by calculating, for example, the Euclidean distances between all data points of the original data in the high-dimensional space. For example, if we were to calculate the Euclidean distance between data points 1 and 5, we would plug in the x, y, and z coordinates in this equation, and do the math. We now plug in the rounded Euclidean distance in a distance matrix. If we calculate the distances between all data points in our dataset, we will get the following values. For example, we can see that the Euclidean distance between data points 2 and 4 is 5.19. Let's put the Euclidean distances based on the high-dimensional dataset here. Next, we determine the value of k, also called nNeighbors. The default value is usually 15, but since we have a small dataset, we set this value to 3. This means that when we consider, for example, the distances between data point 1 and all the other 4 data points, we only focus on the 3 closest points to data point number 1. For example, these are the 3 closest neighbors to data point number 2, and these are the 3 closest neighbors to data point number 3, and so forth. For data point 1, we plug in its shortest distance here, which is its distance to data point number 3. And then we plug in the distance between data point 1 and 2 here. We set sigma to 1 for now, and do the math. Then we plug in the distance between data points 1 and 3. We skip this distance because data point 4 is not one of the 3 closest points to data point 1. Finally, we plug in the distance between data points 1 and 5. If we do the math in the numerator, we will get these values. This is an exponential distribution, where sigma determines the area under the curve. Since sigma is set to 1, the area under the curve is now 1. If you plug in the calculated values in the numerators in the exponential distribution, we can see that the heights of the curve correspond to these values. Sigma is now optimized so that the sum of the heights is equal to the log base 2 of the hyperparameter n neighbors. Remember that we previously set the hyperparameter n neighbors to 3. Sigma is now adjusted so that the sum of the heights is equal to 1.58. If we set sigma to 0.648, the sum will be equal to about 1.58. Note that the area below the curve is now 0.648. Since the area below the curve must be equal to 1 to be defined as a probability distribution, the unnormalized exponential function used in UMAP is instead referred to as an exponential kernel. This is the adjusted VIJs for different optimized values of sigma for each row. Note that every row now sums up to about 1.58. These dots represent the distances that we did not consider because they were not among the 3 closest neighbors. This is therefore a sparse matrix. A sparse matrix is more computationally efficient to work with, because the memory use is n times k instead of n times n. These elements, including data points that are not the 3 closest neighbors, are set to 0. Next, we symmetrize this matrix by this equation, where the symmetric distance value between, for example, data points 2 and 4 is equal to 0.2993. If we do the same calculations for the other values, we will get these values. Note that the upper triangle holds the same values as in the lower triangle, which means that we have a symmetric matrix. These are the normalized distances for the high-dimensional data that will be compared with the distances of the low-dimensional data. Next, we compute the distances for the low-dimensional data with this equation. This equation has 2 parameters that are optimized based on the hyperparameter mean distance that is set by the user. I will explain how these values are calculated at the end of this video. If you set the hyperparameter mean distance to 0.5, A will be equal to 0.576 and B will be equal to 1.363. If A and B are set to 1, we'll get the same expression as in the numerator of the corresponding equation for the t-SNE method. We start by calculating the Euclidean distance between the points in the low-dimensional space. These are our initial random coordinates in 2 dimensions. To calculate the Euclidean distance between, for example, data point 1 and 3 in the low-dimensional space, we plug in the x and y coordinates and do the math, and plug in the resulting value in the distance matrix. If we calculate the distances between all data points, we will get this matrix. We can now plug in the Euclidean distances in this equation and compute the first Q value like this, which we plug in into the Q matrix. If we compute the other values, we'll get the following Q matrix in which the main diagonal is set to 0. These values will be adjusted by comparing them with the values in our previous symmetric V matrix based on the high-dimensional data, by using the following cross-entropy cost function that we minimize with the gradient descent method. We here only need to include the values in the upper triangles. If we compute the value of the cost function based on the upper triangles, we get a value of about 7.7. We'll now change the coordinates of the points in the low-dimensional data and update the values in the Q matrix to reduce the value of the cost function. UMAP uses stochastic gradient descent to do this, but to simplify the calculations, I will here just use the gradient descent method. To compute the gradient descent, we need to find the derivative of this function, which looks like this. We know the values of A and B, and the values in the V and Q matrices, and the coordinates of the points in the low-dimensional data, and the corresponding Euclidean distances. We'll here see how to calculate the derivative based on the first data point. We plug in these values here, and then these values here. Then we plug in the Euclidean distances between data point 1 and all the other data points here. Next we plug in the x and y coordinates of data point 1 and the coordinates of the other data points, and do the math. Note that this calculation is based on more exact numbers of the values in the Q matrix and the distance matrix. If we do the calculations for all rows, we'll get these values, which we plug in into the gradient descent formula, where alpha is the learning rate that we here set to 0.01. These are our current coordinates in the low-dimensional space. So, if we multiply these values by the learning rate and subtract the resulting values from these values, we'll get these updated values. And the coordinates of the data points will therefore be updated like this. Note that data points 4 and 5 have now moved closer to each other, so that the location of the points in the low-dimensional space are similar to the ones we see in the high-dimensional space. With these updated values, we have reduced the cost function from 7.7 to 6.9. If we repeat the same calculations 200 times, we'll get the following plot, which closely resembles what we see in the high-dimensional space. After 200 iterations, these are the values in the Q matrix, which closely match those in this matrix. Finally, I will explain how the parameter mean distance is related to the parameters A and B. The red curve is the target curve that takes the exact shape based on the parameter mean distance. The mean distance controls the length of the flat part of this curve. If the distance is shorter than or equal to the mean distance parameter, the value of the curve is equal to 1. And if the distance is longer, the curve follows an exponential decay function. Now we use nonlinear regression to fit this function to the points on the target curve for a range of different distances, so that we get the following fitted curve, where A and B are estimated to the following values. If we would plug in these values here and change the value of d between 0 and 6, we would get this blue curve. So when we plug in the distances in this function, it can be seen as we compute the heights of this fitted curve based on the distances between, for example, data point 1 and the other data points. If we increase the mean distance from, for example, 0.5 to 1, the flat part of the fitted curve increases, and the distances between the data points increase, so that we get about the same optimal Q values. The points are therefore forced to keep more distance between them when we increase the parameter mean distance. This explains why the data points are more spread out within the clusters when we increase the parameter mean distance. This was the end of this video about UMAP. Thanks for watching.